Why did the plant get sent to the principal's office? It kept passing notes during class. Chlorophylled with secrets. Here's to 3.4 AP Biology Photosynthesis Note Disclaimer.
Let's talk about a brief overview of the term photosynthesis. Photo, which is a term in Greek known as light, and synthesis, which means put together, essentially basically tells us what we need to know about photosynthesis. Photosynthesis is a process in which things like plants, algae, and bacteria utilize the sun's energy to then convert and assist in converting carbon dioxide and water into glucose and oxygen, which ends up becoming a byproduct. So when we talk about plants, let's think about it in the ground. They're going to take in the carbon dioxide that we then release as human beings, and we plant and feed our plants water, so they take in H2O, or we could have natural rain, which is also H2O or water, and the plants release sugars, aka C6H12O6, and just a brief review of Unit 1, remember that goes under the macromolecule group, what? Carbohydrates, and also releases as a byproduct water. The act of photosynthesis is nearly essential for all life. Think about it. We as human beings need to consume plants to sustain ourselves. And at the same time, we also breathe out carbon dioxide. And we can't have too much carbon dioxide in the atmosphere. So plants help ensure that the carbon dioxide is soaked and then also provides us oxygen as well. So in a weird way, we depend on plants a lot. In this unit, or specifically in this section, you're going to be talking about photosynthesis, and you're going to need to understand the specific steps. Keep this in the back of your mind. When you're thinking about photosynthesis, think about it as the opposite of cellular respiration. In a future section, you're going to be covering cellular respiration. And oftentimes when it comes to AP biology or just tests in general, you're asked to compare and contrast photosynthesis to cellular respiration. So make sure to keep that in mind when you're reviewing for a test or a quiz. Let's talk about the idea of photosynthesis and how we got here. First, we need to talk about Units 1 and Units 2 because this will help us understand what we're learning in photosynthesis in Unit 3. Now, please note we talked about the endosymbiotic theory previously in Unit 2, and we're going to bring this up again. What happens is we believe that photosynthesis began with cyanobacteria, which is this item right here, which is considered one of the first organisms to utilize oxygen into the Earth's atmosphere, meaning that this was the organism to release oxygen. What ended up happening is we believe that once oxygen was in the atmosphere, it brought about this idea that different organisms were able to harness the oxygen. If you remember in the endosymbiotic theory, we stated that there were organelles within the cell, such as mitochondria, that have evolved. And the reason why we believe that is eukaryotes are very advanced. And if you remember, the organelles, specifically the mitochondria, which kind of is the powerhouse of the cell, has its own relative DNA and is its own entity where it sort of is like a mini cell within the cell. And this gave molecular evidence alongside with other fossil evidence to further establish this idea that an oxygen-rich atmosphere was necessary for establishing more evolved organisms. Through that theory of endosymbiotic relationships, we now understand that photosynthesis and the pathways extend to eukaryotes such as plants and algae. And this is really important because what happens is we now realize that plants can harness the CO2 and produce oxygen alongside with the taking in of water and the sun's energy to flourish and basically help other organisms survive as well that need to utilize the oxygen because they themselves produce that oxygen. So it all revolves around each other. Now we know that there are three types of troughs or organisms. First is an autotroph, which basically takes organic molecules and CO2, and what happens is they create their own organic molecules from the CO2 and inorganic materials. So those are autotroph, auto meaning self. So they create their own organic molecules. Photoautotrophs, you notice the root word photo there. Photo meaning light. So it uses light energy to produce the organic molecules or compounds. And if you notice photoautotrophs, so photo meaning light, auto meaning self, so it sort of connects back to autotrophs in a sense. And then we have heteroautotrophs. Hetero, you could think is utilizing like hetero Homo sapiens. So we can think about ourselves in the terms that we rely on autotrophs for both food and our oxygen to sustain. So these are the three main types that exist and have evolved along the process of photosynthesis being used.
Now meet the photoautotroph, a solar-powered genius. It catches light, flips it into fuel,
and finally, the heterotroph. That's us. We rely on the creators for food, oxygen.
Hi, I am the thylakoid membrane, and I am where the light reaction occurs. In this table, you'll notice the features that are involved in the light reactions, which we will discuss in this video. Unlike my friend, the thylakoid membrane, yes, we are both in the chloroplast, I, the stroma, have a special process known as the Calvin cycle, which is known as light-independent reaction. So just be aware of that. In this video, we're going to get into more details about the photosynthesis steps. However, for now, we're going to first cover what the chloroplast is and a brief overview of what goes in and what goes out in terms of photosynthesis. First, let's take a look at this picture right here. You notice the green pigment of this structure. This is known as the chloroplast. The chloroplast is where the site of photosynthesis occurs. There are two main steps in photosynthesis. The first step is known as the light reaction or the light-dependent reaction, and the second step is known as the Calvin cycle or the light-independent reaction. When it comes to the chloroplast or the site of photosynthesis, this includes several key steps. I want you to understand this green pigment here and remember what leaves look like. The thing is, chloroplasts are found in the mesophyll cells of leaves, which is the reason why you see this green pigment. But you see it because what happens is there's something known as chlorophyll. And chlorophyll is the green pigment that captures the sun's energy. So when we talk about the sun's energy, we're talking about the first of the two steps, which is known as the light reactions. These light reactions occur in what's called the thylakoid. So you see the thylakoid or this disc-like shape. So this is a disc and this is a disc. The thylakoid is a disc-like shape because it needs to be flat so that way it can capture as much of the sun's energy. It contains that chlorophyll. A group of these thylakoids are known as grania. And then what happens is there's a fluid-filled space, which is where the second step of photosynthesis occur, which is known as the Calvin cycle. And this fluid step, or I should say the Calvin cycle is the step, the fluid-like space is called the stroma. And also when we're talking about light reactions, we're going to have a process where there's ATP production, aka the energy, and that occurs in the thylakoid membrane. So these all are interconnected together. Now that we've spoken about the chloroplast, let's get back to this photo right here. Overview, we have the sun's energy and some water, which goes into our plants and starts up the process of the light reactions, which, by the way, occurs in the thylakoid membrane. So think that disc-like shape. And that disc-like shape is there to capture the sunlight, so it can help produce several things. The first thing is, as a byproduct, we have O2. So O2 is because the water, if you see hydrogen and oxygen, the oxygen splits off because it's going to use this hydrogen to then create what we now know as NADPH. So there's NADP, and then that hydrogen attaches, which now gives it NADPH. So in the light reactions, you have the byproduct of oxygen, and you have some NADPH, and you also have some ATP. The ATP and the NADPH, I should say, both are carried into the stroma. Remember, the stroma is that fluid-like sac in which the Calvin cycle occurs. Please note, alongside with the NADPH that's produced from the light reactions and the ATP, we have an outside product of carbon dioxide which comes in. The goal is to take the carbon dioxide and convert it into what we consider G3P, which is considered a sugar. So what ends up happening is you have this production of sugar which is released, and we're going to talk about that in more detail. And then as leftover, we have ADP, adenosine diphosphate, a phosphate, and NADP, which all go back into the light reaction. So this is very much a cyclic activity. In the rest of the videos, we're going to be talking about photosynthesis more in detail, as there are specifics in the light reaction you need to know and in the Calvin cycle that you need to know. But for now, you know that the green color is because of chloroplast, and the key structures that are important include the thylakoid, which disc-like shape is important to capture that sun's energy, along with water, which allows for splitting of the water, where oxygen is a byproduct that is released in the light reaction. However, together with the light and the H2O and the NADP and ADP produced from the Calvin cycle, we have production of NADPH and ATP from the light reaction, which then churns the Calvin cycle alongside with outside CO2 in the stroma to then produce sugars, which are released. So that is a brief overview of the steps. Now, before we go into more details, I wanted to go into the chloroplast and that color chlorophyll. The thing you need to understand a little bit of physics is the visible spectrum here. Here, the thing is, light is what is going into the chloroplast, right? And it moves as waves. Remember, the distance between each peak of these like waves is called the wavelength, and the complete range of these waves is therefore frontally known as the visible light spectrum, which ranges from 380 nanometers to 750 nanometers, which is crucial for life. And you might have heard the term Roy G. Biv, red, orange, yellow, green, blue, indigo, and violet, and that relates to the wavelengths here. So for example, the higher the wavelength, like for example 800, you get that red color, and then you transition to orange when you're in that like middle range, where eventually you get into violet, hence the term Roy G. Biv. So red, orange, violet, green, blue, indigo, violet is the color arrangement. So you can see it goes from red to Biv, which is violet, all the way on the one side. So essentially, the more red, the higher the nanometers or the wavelength, essentially. And you can see the wide peaks between that red color there. And why you might need to know this between light and visible light is because you can get a question where it asks you, is the light being transmitted, reflected, or absorbed? So what you need to know is that substances that absorb visible light are known as pigments, and the color that we see on the outside is actually reflected, and what's being absorbed is the process of photosynthesis. So let's explain more in detail. So if we think about chlorophyll, so chlorophyll is going to be that green color, and I want you to think of carrots or carotenoid. What color are carrots? They tend to be orangey color. So if you have orangey color, they're going to be between the 570 to 670. So 570 to 570 to, you want to say 670, like 670-ish. This range right here would be where you notice that there is starting to be least absorbance. However, the peak of the absorbance is somewhere around 400 to 500 for this carotenoid color. And so this is important because if you're given a graph or if you're shown a picture, you now know that the higher the absorbance, that is going to be equivalent to what you do not perceive. So if we're absorbing that and the plant is absorbing that color, so that's the 400 to, I think it's 500, we don't see this color for this orangey color of carotenoids. Instead, we see what is not being absorbed because that is what's being transmitted. The color we perceive is what's being reflected, and what's being absorbed is the utilization through photosynthesis. So let's take a look at the green color. So the green color, if you could see here, is between 500 and a little bit less than 500, so like 460 all the way to around, give or take, 560. So if you look in the green color here, what's being absorbed is the 400 to 500-ish, and then it starts peaking again around 700, which is related to these colors right here. So what's being absorbed is not what's being seen, and what is then being reflected, or the lowest, is what we do see. So you can see that corresponds to the wavelengths of 500-ish is corresponding to that green color. And this is important for chlorophyll because in plants, absorbing all colors of light except for green, which is reflected, is what gives plants that green color. And the visible light spectrum plays a really key role in photosynthesis by ensuring that we're delivering energy in the form of that absorbed light. So what's being absorbed is what's utilized in photosynthesis, and what's being reflected is what's not being absorbed, usually the one that is not peaking. And so this is important, especially if you're given some sort of free response question or multiple choice question. You now know if they're trying to alter the wavelengths to note that the ones that are being absorbed, or the ones that are in the peak, are not going to be the color you see. Instead, the one that is the dip, like the 520 to 540-ish range, matches or aligns with the color that we can see from the visible spectrum. So this is how you can distinguish for a free response question if you are given something like this. Now let's go into more details when it comes to the stages of photosynthesis. Now there is more specifics with photosynthesis, but I want to give this to you as a preview. This is the chemical reaction that is noted for the light reaction. Remember, photosynthesis is a process when we're capturing the light, and we store the energy that is the form of glucose, which is being released from the Calvin cycle. The chloroplast structure, especially these thylakoid membranes, are there in a disc-like shape to ensure that we're maximizing that light capture, and we are producing what's known as ATP ATP in this process to help generate the Calvin cycle. So when we think about like reactions, remember it is in the thylakoid membrane. We are harnessing the sun's energy to produce what is known as ATP and NADPH. Remember, oxygen is being released because we're taking the hydrogen and placing it to the NADP that was previously provided from the Calvin cycle. So you see that NADP that's coming in. And we are creating NADPH because the water that came in that is attaching to that hydrogen. The oxygen is then being released in this process into the atmosphere. And we as human beings could utilize that oxygen. That's why plants are beneficial to us as human beings, because it provides us oxygen. Now, if we look at the light-independent reactions, which is the second step, which occurs in the stroma, remember we have carbon dioxide that comes in from the outside, that NADPH that's being produced from the light reactions in ATP to then turn the Calvin cycle, which produces some glucose, NADP plus, and ADP. For the Calvin cycle, or the independent reactions, I should say, which don't depend on light, there are three main phases. Carbon fixation, which utilizes the CO2 from the atmosphere, reduction, and regeneration of what's known as RUBP. The purpose of the regeneration is to produce G3P, which is a form of sugar. So essentially the goal is to make glucose, or a type of sugar. So that's a brief overview. In the next part, we're going to go into more details about the light reaction and the Calvin cycle.
The light hit me!
If you notice this visual here, you notice a very complicated visual, and we're trying to break down what happens in the light reactions, and we're going to do that in 2 slides that you see here. Please note this is a more detailed slide and this is a less detailed slide, but both of them basically cover the same thing. We're going to cover the process of the first step in photosynthesis, aka the light reactions, in detail, and then we're going to cover it again, now noting the basic language that we have understood. I want you to understand that what is happening here in this image is the same here as it is here. Both of these demonstrate the same process of the light reaction. They're just demonstrated in 2 different ways. And this is just to give you a very unique idea about the light reaction. So let's first cover the shape of that disc. You remember what that disc is called? That's called the thylakoid membrane. And the purpose of that thylakoid membrane, or that disc-like shape, is to ensure that we are able to capture or harness the sun's energy and convert it to chemical energy. So you'll notice here there's light hitting. That's the symbolism of yellow here. And you notice the light here. This is just what the symbolism of yellow here symbolizes. It's that conversion of light energy into chemical energy. Remember, energy can't be created nor destroyed. It's just converted. So what's going to happen is you have the creation of ATP and NADPH, which are the energy conversions that we have here. Our initial inputs include light and water, H2O, and their outputs are going to be that ATP and NADPH. Remember, we have ADP that's also coming in as well, and NADP plus from the Calvin cycle, which is cyclic. But for now, let's start this in a linear format so we can really hone in the process. First step that we're going to talk about is the water. The water is going to hit with the light, is going to react and formulate water splitting. The process of water splitting itself is called photolysis. Photolysis. And photolysis, basically, you can see the term lysis here, which means splitting. So we're splitting with water. The act of splitting the water is going to take that H2O with our light. So photo means light. So splitting with light, we're taking that H2O, we're taking that H2O and we're splitting it into oxygens and hydrogens. We're splitting it into oxygens and hydrogens. Remember, these are two of the same photos. They're just showing very different things and different images of the same things, I should say. So these are just H2O, H2O. We have splitting of that water. We have the oxygen and the hydrogen. Remember, oxygen is a byproduct. So oxygen is going to be released in this process here. And then what ends up happening is we have the hydrogen ions, which are going to be utilized in that thylakoid lumen. So the thylakoid lumen is that inner space. You can notice that there is a bilayer here. There's a bilayer here, and remember the phospholipid bilayer. The chloroplast, specifically the thylakoid membrane, is embedded with that bilayer as well. And the bilayer has these specific, if you notice, proteins that are going to allow for the electron flow. And that's what we're talking about more in detail. It's just more simplistic with this image here. So to recap, we talked about photolysis, which is the act of water splitting. And once that light hits water, what ends up happening in the thylakoid is we have water splitting into oxygen, hydrogen. Oxygen is then dispersed into the atmosphere, which we as human beings utilize. And hydrogen is released into, you could see the thylakoid lumen right here, and is being used further on down the line. Now, here is the second step. Essentially, we have or simultaneously, I should say, is the sunlight, which excites the electrons. This is going to first occur in photosystem 2. And you notice it's green here to symbolize chlorophyll. So it's lighting up that chlorophyll. And what happens is, remember, this is happening simultaneously. This is happening simultaneously as we're hitting that water. And it's going to hit what's called P680, which is going to be the excitation of those electrons. So we have excitation of those electrons. You can see the little E's here. They're very excited. And photosystem 2, aka P680, is the first step as compared to photosystem 1. I say as compared to step 1 to step 2, meaning photosystem 2 comes first, then photosystem 1. But remember, this is cyclical. So what I mean by that is they're happening simultaneously. So we essentially know that the H2O goes into photosystem 2 first. And then what that does, that electron flow will then need to go through these series of steps here, which you don't really see in this picture here. So I went ahead and I posted out the electron flow overview so you understand more about the steps in the electron transfer chain 1. Now I say 1 because after photosystem 1, 2, there is also an electron transfer chain as well. Essentially what we have is water going into photosystem 2, that's PS2. Then we have the primary electron acceptor, which excites that whole process of P680, the chlorophyll, hitting with the light. And that basically triggers the electron transfer chain, which leads to this whole process. So you see, I can't pronounce it, but it's pheophytin, which goes to plastoquinone. And then you have cytochromes, which hit plastocytin, and then go to photosystem 1. Now, the specifics of these names, do you have to know? I wouldn't really say you have to know these specific names. You do need to know photosystem 1. Photosystem 2 goes to the electron transfer chain and then goes to photosystem 1. However, for AP biology, the test has been getting progressively harder. And because of this, I try to allow students, if they want to understand the specifics, to do so with the electron transfer chain. Just understanding that there is these steps before it hits photosystem 1 helps some students really understand the difference between photosystem 2 and photosystem 1. So once we have photosystem 2 and we have the electron transfer chain, which has all these various processes that it goes to, we have production of ATP. So in the act of photosystem 2 to photosystem 1, remember, key importance is that you have ATP production. And I want to circle back to this photo here because that ATP production is conducted during the ATP synthase. So ATP synthase is what really drives ATP production via taking the hydrogens and passing them along, those ions that we have from water, and passing them along this ATP synthase. That production of passing the hydrogen along essentially triggers ATP production, which is what's happening in ETC1. We don't call it ETC1, we just call it ETC, but I'm going to say between photosystem 2 and photosystem 1. That is what's happening in ATP synthase. And we also call this chemiosmosis when we're generating ATP and NADPH through photophosphorylation. So this act of generating ATP and the electron transfer chain of NADPH, which we're going to cover in just a second, is also known as chemiosmosis. So I wanted to preview that. I'm going to go back to that in just a second, but I wanted to let you know that ATP and NADPH production, which are both energy, with the assistance of light, aka photophosphorylation, is also known as chemiosmosis. Now, going back to the photo, now we have photosystem 1, right? We have photosystem 1, which is what we're covering here. So photosystem 1 is going to hit P700, and what is happening is those electrons are now being re-energized. And then what happens in photosystem 1, you can see the trail here, we go to FD, and then we have NADP reductase. So NADP reductase is sort of like ATP synthase. And what happens is NADP reductase, if you notice the ASE ending, and if you remember from Unit 1, or I think Unit 2 into 3, we did talk about enzymes, and the ending ASE is a trigger to let you know that that is an enzyme. So what's happening is, remember, we have NADP from the light independent reactions, aka the Calvin cycle. Remember, that's coming in. What's happening here is you have, just like production of ATP, which has ADP coming in. So you had ADP coming in from that Calvin cycle to help convert to ATP. You also have, which we're going to cover in a second, you have the second electron transfer chain simultaneously taking NADP and adding the H to then create NADPH. That's the NADPH formation. So now we covered step 1, step 2, step 3, step 4. We did talk about step 5, but that we said was step 1. So it really depends where you want to look at it. If you want to start here and go around, or if you want to start with the water and go around, either way, they're all the same process happening simultaneously. Now I want to cover this process here and this process here, both electron transfer chains. Remember, we have ADP, which is coming from the Calvin cycle, and we have the NADP, which is also coming from the Calvin cycle. And the introduction of exciting the electrons is what allows it to cover to ATP from photosystem 2 to photosystem 1, and then photosystem 1 to NADPH is what is essentially converting it to NADPH. The act of generating these is through the photophosphorylation, which is the excitation of that light by using a protein gradient. So NADPH is a protein gradient, and ATP synthase is also a protein gradient. And the act of the protons, all of these hydrogens in here, pumping and transporting into that thylakoid lumen is going against the gradient. So they're constantly pumping against the gradient. And this is what is backflowing in the ATP synthase, which is allowing for the ADP in that electron transfer chain in photosystem 2 to photosystem 1 to convert to ATP. And this is also a key component in the opposite of cellular respiration. Remember, we're covering photosynthesis in this section, but we're also going to be covering cellular respiration in a different section. And a lot of the test questions will ask you to compare and contrast the two steps. And one of the key similarities is you'll notice chemiosmosis and utilization of ATP synthase. So that's just important to note if you get a trick question. Now, if you notice in these steps, I talk about it sometimes as water step being number one, and sometimes I might say step number one is sun excites. Know that this is like a cyclic process. So what I mean by that is it can go back. You could start here and then go back as long as you're doing it in the correct order. And here to generate or to better understand, using photosystem one by itself to just produce ATP is sometimes considered cyclic because it's just producing it by itself. However, it's considered non-cyclic when we're producing photosystem 2 and photosystem 1 to simultaneously create ATP, NADPH, and oxygen. So just being aware of the process of creating ATP can be done solo, or it can be done in production of NADPH and O2. Now, to further cover this along the line, I have created a further table. This covers the same things that we just said. So if you notice here it mentions oxygen released at the very end, which is the same thing as we're starting off in this process. The steps are exactly the same. It's just starting off with photosystem 2 as the initial process, and that light hitting then triggering, which it does, the water splitting. So you can see that here. And so technically it's saying it's splitting, it's going through all these steps, and then we have oxygen release. Either way, you know that light simultaneously hitting with water is going to trigger these steps here. And also remember the goal is to release O2. Chlorophyll is what is absorbing the light. For photosystem 2, it's happening at P680 and then at 700 for photosystem 1. ATP via chemiosmosis is called photophosphorylation when we're talking about plants. However, in cellular respiration, when you're going to talk about it in a later section, it's called oxidative phosphorylation, which is in cellular respiration. So the process is very similar. However, what's happening is the naming convention is a little bit different. Now let's talk about more specifically the electron transfer chain. This is just showing more details about that process in specifics. So you'll notice here that we're talking about the step of the thylakoid membrane. We're just generating this idea that we're producing ATP and NADPH. The goal is to reduce NADP, which is going to accept the hydrogen from that lumen that contains all those hydrogens. This is just showing more details about the ATP synthase and the act of the ADP attaching a phosphate with the churning of the hydrogen through that ATP synthase. So this is a more detailed photo. You can see the bilayer here. This is the lumen that we previously talked about. And we see the light hitting. We hear it, we see it hitting at P680. This is supposed to show photosystem 2. And the box here just indicates more specifically what the electron transport chain really is. And it is that process of allowing the electrons to travel through, and it's embedded in that thylakoid membrane, similar to the phospholipid bilayer. So you see the shape, it looks exactly the same. You see the lipids and their bilayer formation. So these are embedded and it's exciting the electrons in that electron transport chain. The goal is to then create these hydrogens that you see here within the thylakoid space, creating some sort of protein gradient, which is going to force the ATP synthase to pump, constantly pumping and creating a protein gradient. That act of flowing back into the ATP synthase is essentially driving this ATP synthase in chemiosmosis and is taking the ADP to ATP, attaching that phosphate with the addition of the hydrogen pumping. I also provided the electron flow, so you have more details. It's the same thing that we showed before, and this is what's creating that ATP. Now, in the production, we have ATP and NADPH. This is going to be carried out in that second step called the final Calvin cycle or the light independent reactions. Remember, we have have the hydrogens also simultaneously, not only producing ATP, but we have NADP, which is shown here, which you see there is the hydrogen attaching to formulate the NADPH. So all of these images are exactly the same. It's just showing it in a different way or format in case you're seeing this on a test or exam, because can be a difficult topic to cover. Okay, now let's go into chemiosmosis more in detail. We have the protons, the hydrogens that are pumping down against their concentration gradient, moving from the lumen into the stroma. You see that it says stroma here. And this act is taking ADP adenosine di meaning two, phosphates attached to make adenosine three phosphates. So what we're trying to say is that we're going to a process called photophosphorylation. Photo meaning light, phosphorylation meaning adding a phosphate. So this whole process is called photophosphorylation or chemiosmosis because we're utilizing light to pump against the gradient to produce ATP. This powers the next step, which is called the Calvin cycle or the light independent stage.
I'm running because I'm about to break down. Here I go into the Calvin cycle.
Now let's talk about the second stage of photosynthesis, which is also known as the Calvin cycle, also known as the light independent reaction, which means that it does not depend on light in this step. Now, in this first step, you're going to notice that CO2 is the aggressor or the starting process in the Calvin cycle. The way I like to think of it is Calvin begins with the letter C and so does carbon dioxide. Essentially, there are three main phases in the Calvin cycle, known as carbon fixation, reduction, and regeneration of RUBP. We're going to cover these three main steps in detail. Please note you don't need to know as detailed as I am telling you in this video, but it's really helpful when you're trying to get that 4 or 5 on the AP biology exam. So I am covering a little bit more details than what you're expected to know for the exam. Let's first cover step 1. So step 1 deals with carbon dioxide, and one carbon dioxide molecule will combine with a 5-carbon sugar known as ribulose biphosphate. And if you notice that ending there, OSE, if you think of glucose, fructose, galactose, those are all carbohydrates. And carbohydrates are a type of sugars. And if you remember from Unit 1, your macromolecule section, that ending OSE is a big indicator to let you know that we're talking about a carbohydrate. In this case, a 5-carbon sugar. So each CO2 will bind to that 5-carbon sugar. And you'll notice here in the clips that there are these black circles. I want you to think of those as carbons. Do you need this detailed information for AP biology? Not really, but it helps you understand the picture more specifically. So if you look at this structure right here, you could see the 5 carbons with the 5 black dots. And I want you to remember that. Now we have ribulose biphosphate or ribulose 1,5 biphosphate or RUBP. So this is known as RUBP. And this is what we're talking about, the 5-carbon sugar. You could see the 5 carbons there. And you might notice it says 1,5. 1,5 is just letting you know that there's attachment at the 1 carbon, at the 5 carbon. This is a little bit more advanced, but I just wanted to point that out, why they call it ribulose biphosphate 1,5, why the 1,5 is there. It's just to show the position. It's the same thing as saying RUBP. So you have this RUBP structure, And this RUBP structure is going to connect with carbon dioxide. Now, you notice this structure here, or this term right here, that is called rubisco. Rubisco. And if you notice rubisco, I want you to think Nabisco. And if you think of Nabisco, you think of sweet treats. And I always think of chocolate chip cookies. Well, rubisco is an enzyme. And what it's going to do is this enzyme is going to come together to essentially facilitate the CO2 and the RUBP combining together. So the rubisco comes in as the enzyme, and remember, it's trying to get reactants to products because that's what enzymes are. We learned that in the end of Unit 2 into Unit 3, we touched upon enzymes, and if you remember, enzymes are basically lowering the activation energy and allows reactants to go to products. So our goal is to try to take this RUBP and the CO2 and combine to create a reaction with the assistance of rubisco, which is our enzyme, which is going to lower the activation energy. See how it all ties in together? So now the result or the product is going to be an unstable 6-carbon intermediate split. So what that basically means is your RUBP, which has the 5 carbons here and the 1 carbon here, is going to combine and split into what we now consider 3-phosphoglycerate. So let me explain more in detail. So remember, you had the 5 carbons here and you had the 1 carbon there. So that makes 6 carbons, right? So they combine together, and 6 divided by 2 is 3. So you have 3-phosphoglycerate here and 3-phosphoglycerate here. So you have 2 molecules here. So what you need to note is that for every 1 carbon dioxide that starts off a reaction, what ends up happening is you have a molecule that splits with RUBP. So carbon fixation is about taking carbon dioxide, taking the RUBP, combining them together, and then splitting them equally. So that creates an unstable 6-carbon intermediate, which it allows it to split. So that's where they're saying there is unstableness. You can't just have 6 carbons attached together like that, and you don't need to know the reasoning why. It's just unstable for now that you need to understand, and that unstableness produces this split, which gives way for the 3-phosphate here and the 3-phosphate here. So this whole process from here to here is your first step of carbon fixation. So that's your first step. This is where carbon enters into the cycle, and then for every 1 carbon, to reiterate, you get 3-phosphoglycerate. And you get 2 of these 3-phosphoglycerates. Now, why this is important is before we gravitate towards that second step is I want you to notice the 1, 2, 3-phosphoglycerates. So you have 1, 2 for every 1 carbon. The 1 of the 2 phosphoglycerates exits the process and just continues on with different pathways. It's not necessarily needed for the Calvin cycle and therefore just disappears essentially. Now, during the second step, which is called reduction, I want you to think reduce. I think you want to understand getting smaller. What happens is you have 1 G3P, sorry, 1 PGA or 1 3-phosphoglycerate. So when I say PGA, that means phosphoglycerate. That will go ahead and energize this ATP. This starts the process of reduction. This process of reduction, by the way, that has the ATP come in, do you remember where this is coming from? This is coming from those light reactions. Okay, so if you remember in the light reaction, we produced ATP. That light reaction's ATP is going to come into the Calvin cycle alongside with the 3-phosphoglycerate or PGA, and it's going to formulate what's called 1,3-phospho biphosphoglycerate, or this structure you see here. Important to note that ATP has now reduced, hence the term reduction, to ADP. Adenosine triphosphate, adenosine diphosphate means that a phosphate has been removed. Now, you might be thinking, where did that phosphate go? The phosphate was used to energize this structure right here. So now that's why it's called 1,3-bi meaning 2, phosphoglycerate. So what you see there is the term phosphate. And before we had 3-phosphoglycerate, right? And you see that it's just one phosphate. It doesn't show it in the structure, but by the naming convention, you could see here that it says phospho, meaning one phosphate, and the adenosine diphosphate removed a phosphate from adenosine triphosphate, which came from the light reaction, and attached to that phosphoglycerate, the 3-phosphoglycerate, to then form 1,3-bi meaning 2 phosphoglycerate. So there's 2 phosphates instead of 1 coming from that ATP structure. So that phosphate attached to produce 1,3-biphosphoglycerate. So essentially the reduction process allows for phosphate to be removed and attached to 3-phosphoglycerate to then produce 1,3-phosphoglycerate. So that's the first part of reduction. Now, also coming from the light reaction is NADPH. Okay. Now, what's going to happen with the light reactions, NADPH, it's going to take that 1,3-biphosphoglycerate and it's going to go in and churn the cycle, I should say, to reduce NADPH to NADP. So there's a missing hydrogen. Where did that hydrogen go? That hydrogen produced, now taking the 1,3-biphosphoglycerate, and created what is called glyceraldehyde 3-phosphate, or G3P. Some things to note when we're talking about this is we had one CO2 come in, right? We had one CO2 come in. And for every one CO2 come in, they have this constant cycle that comes in. So I want you to think that there are 3 CO2s that come in essentially, and so this is going to be tripled. So for every 3 CO2s that come in and enter the cycle, you have 6 G3Ps that are produced. Now, you might be thinking, well, this is a random number. It doesn't come from anywhere. I want you to focus back to Unit 1. Remember what glucose looks like? Glucose looks like C6, H12, O6. Remember, we're producing sugars. Sugars are this structure. This is where Unit 1 comes in. Carbon dioxide has 1 carbon in it. So I need 3 CO2s to come in cyclically to then double to make 6 G3Ps. So what you need to know is for 3 CO2s, we're going to produce 6 G3Ps. Okay. And what ends up happening is 5 of these G3Ps are ended up recycled. And that is where regeneration starts. So what happens is you have 6 of these G3Ps, and 5 of the G3Ps are going to end up being recycled to regenerate RUBP. So RUBP is up here. So we need to note that 5 are going to be sent this way to create RUBP. And one of these G3Ps are going to then be released. And the one that is being released, remember there's 3 carbon here, is going to end up building glucose because it's going to end up being released. So if you think about it, you have 5 that's being recycled, and then only one G3P is going to assist in facilitating the 6 carbon that's necessary for glucose, so or the sugar. So you need to go through it twice to produce essentially one sugar, if that's what you're producing, one glucose molecule. That's just a little side note, but for now, when we're ending reduction, what we're saying is that out of the 6 G3Ps, we're sending 5 to recycle over to create the RUBP from this molecule, and 1 is being released to then create this sugar molecule, and that is going to be released as a product. Now, let's talk about that third step of regeneration. Now, remember we have the 5 G3Ps from here. That is going to be rearranged to formulate our UBP with the addition of ATP. So what I mean by that is essentially we have ribulose 5-phosphate, and we need to add additional phosphate to create the RUBP. So the end product that we want is RUBP, right? We want that by 2 phosphates. But just like before, we only have 1 phosphate here, and the G3P needs to come in and take the ATP, also coming from the light reaction, take that phosphate off, and attach it to the ribulose to become ribulose 1,5-phosphate. So in regeneration, the final step, you're basically taking the carbon structure of the 5 G3Ps and you're back into the 3 molecules of RUBP. So this process requires the ATP to regenerate into RUBP, which is crucial for the next step in starting the cycle again to create more CO2. So in phase 3, or that final step, we have the 5 G3Ps. Remember, 1 is being sent out to create sucrose, and the 5 G3Ps, so the 5 G3Ps, yeah, the one that went to glucose. And then what ends up happening is you have this idea that you are taking the RUBP and you are creating it by taking that ATP and to rearrange and create and formulate the RUBP by reducing that phosphate off, taking it off, and adding it to create ribulose 1,5-biphosphate from ribulose 5-phosphate. So that takes 5 G3Ps. So that process is called regeneration. So this is the process of the Calvin cycle that exists. Now, we're going to talk about 2 different types of plants, and these are called C4 plants and CAM plants. I want you to think about dry conditions where you don't have a lot of plant growth. In these environments, what's happening is you have to have plants that are able to sustain environments that otherwise you can't do so. This is a picture of mesophyll cells, and this is a picture of bundle sheath. And what's happening here is the CO2 is grabbed in the mesophyll cells, and this is done by what's called PEP carboxylase. So you can see that ASE ending there, and carbo and oxygen, because that can help you remember. The point is to add the CO2 to the PEP, and what ends up happening is you create oxoacetate. And in this goal of creating oxoacetate, which is then created into what's called malate, is shuttered to the bundle sheath. So the bundle sheath will now have the CO2. And what ends up happening is the high light low or the high light, so like the high light meaning from the sun, and the high temperature means that you have low water content. You want to think about that desert environment. And what happens is you're enhancing the sugar production to compensate so we could thrive in those hot, sunny environments. And the cost is that you're basically utilizing extra ATP that you don't normally have, or energy to take the CO2 and fix it. And what essentially is going on is you're going to have faster photosynthesis with high productivity. However, you have a cost, which is the ATP. So that's what C4 plants do, which is great for favoring environments that have those CO2 need to, if you think about it in those hot, arid conditions, you're not going to have a lot of plants there, which means there's not a lot of excess O2 being released. So you have to favor the CO2. Now let's take a look at cam plants, which favor the stomata opening and closing. So the stomata will open at night, and this is to capture the CO2. You have to think at night it's going to be a lot cooler than during the day. And this is so that way when we open it, we're having that gas exchange. The CO2 is then going to be captured by the stomata. And what happens is during the day, it converts these acids back to CO2 for the Calvin cycle. So basically, you're capturing the CO2 for the Calvin cycle at night to then process during the day. The point is, if it's doing it at night through the cam plants, you're conserving water because you're utilizing cooler nighttime conditions. If you think about when you wake up in the morning, there is a dew sometimes on your grass, and that's because of the nighttime conditions, which are a lot cooler. So we are conserving water by not using up the process of the Calvin cycle. So this is a timing-based plant. So CAM plants are based on timing. C4 plants are based on location. So that's the difference between the two. Now let's take a look at this quiz question. Pause this video, make sure to unpause once completed, and let's take a look at this quiz question together. It says, during the light-dependent reactions, a chloroplast is experimentally modified so that the thylakoid membrane becomes permeable to protons, H+. Which of the following most directly result from this change? So the best answer, did you select it? Let's see. The correct answer is B. Decreased ATP synthesis. Let's talk about it. So remember, we have oxygen that is considered a byproduct of the light-dependent reactions in a normal condition. But because of this experiment, it becomes more permeable to protons. Remember, during the light-dependent reactions, we have the ATP synthase, or the ATP synthase. And the process of the ATP synthase is to go against the gradient. Now, if we're going with the gradient, what ends up happening is it's going to become permeable. So what ends up happening is there's going to be decreased ATP synthase. If we have the thylakoid membrane, so we have the thylakoid membrane, and we don't need to utilize the ATP synthase. It's just going to easily follow through. Now, if we have the ATP synthase that we normally utilize, we're pumping that hydrogen against the gradient, which is beneficial because then we're producing more ATP. What happens with this experiment is we're saying that we're becoming more permeable in this process right here without utilizing ATP synthase, which would be an issue. So that way we would not have against the gradient membrane, which means we would not need to utilize the ATP synthase. So we would have decreased ATP synthase. And going against the gradient drives the process of chemiosmosis. So we need that to ensure that the electron flow continues. So we don't have increased NADPH either. So that's part 3, or answer choice C, why it's incorrect, is because remember, production of NADPH is not really has anything to do with protons passing through. They were talking about light portion rather than the dark portion, right? So light reaction, if it has increased NADPH, right, which it's going to flow rapidly through the ETC. If you remember in that process, the NADPH production processing through the ETC is correct. However, it is before we're talking about pumping the protons. So this, while they're congruent information, it's not the best answer choice. And no effect would not be the scenario because we're changing something experimentally, which alters the ATP synthase. Now, before you click off on this video, just a brief reminder, make sure to check out our courses. We have a full list of videos for units 1, 2, and now 3. We're going into 4, and we have some videos in 5, 6, hopefully to be completed throughout the year. So making sure that you go through our course section, review all those videos in case you didn't understand this video, as they're broken down by section and will be throughout the course of the years that these videos are being posted. Hope this helps.
Appreciate you riding along for our cosmic journey. If today's mission brought you the knowledge that slaps, make sure to hit that like button to boost that signal. Subscribe if you're locked in for our next launch. And don't miss the link tree, it's got all the goods for AP Bio, PSAT Math, and FSAT Math. Before I sign out, make sure you're all in one orbit and diving in. Killing out. Till next time.